Good morning. It's 10 o'clock here in Western Europe. We are going to start our PIGWEB workshop entitled Application of Non-Destructive or Censoring Techniques for Animal Science. This workshop is part of the PIGWEB project, and which is about to finish this month. My name is Jaap van Milgen. I'm the coordinator of this project. Next slide, please. A couple of house rules. So please mute your microphone when you're not speaking. And we'll have time at the end of the whole session to ask some live questions. But if you have questions after each of the presentations, please indicate your question using the chat function so that you will not forget your question and that we can come back to these later on. During the discussion later on, if you would like to speak, raise your hand, please. And as you've noticed, recording is in progress because we like to disseminate this information later on. So if you don't want to be seen on the video, don't put your camera on when you intervene during the presentations, during the discussion. We have 6 topics today, 5 speakers: Cecile, Bonnefont, Örs Petneházy, Claudia, Kasper, Maria, Furnols, and Tamás, Donkó. They will have the different presentations for the 6 topics. Next slide, please. So we have the 6 topics. The first one is by Cecile about NIRS and carcass composition. Then Örs will talk about special anesthesia for imaging. Claudia will talk about the use of DXA in pig production. Maria about computed tomography applied to live pigs and carcasses. Then NMRI examinations of pig carcasses will be presented by Tamás. And then Maria will come back again to talk about pig carcass grading in Europe and classification devices. And then at the end, we have some time for questions and answers in the live session. Next slide, please. So the first talk is about NIRS and carcass composition by Cecile Bonnefont. Cecile has been an assistant professor at France's National Research Institute for Agriculture, Food and Environment, better known as INRAE, and at the INP Agro-Toulouse, which is the engineering faculty of life sciences at the Institut National Polytechnique de Toulouse. And she's been there since 2012. She teaches pig production with a special focus on pig nutrition, and she supervises training of Master 2 students specializing in animal production. In her research, she worked on product quality and fatty liver in ducks by metabolomics approaches, and then she moved on to pig production to study the metabolism of piglets at the end of gestation. She has recently started working at the farm level to study the PDO of Noir de Picor pork, which is a local breed, and the utilization of resources in France's organic farming sector. Cecile, it's all yours.
So thank you, Jaap. I'm just going to share my screen. Okay, can you see it?
Yes, it works.
Okay, perfectly. So good morning, everybody. So thank you for this introduction, Jaap. So I will talk about NIRS and carcass composition. So the objective of my first talk is NIRS a good device to estimate carcass composition on pigs? First of all, we have to explain what is NIR. So NIR is for near infrared spectroscopy. And so it is about the light, or maybe I can just add a, like this, so you can see. So it is about the spectra of the light, and so in near infrared. So when we use a light in a sample, after we will have a reflected light. And this reflected light will depend on the characteristics of the matrix or the samples used. So here on the right, you can see that for example, for water, the water will absorb the light at different wavelengths. For example, around 1,400 nanometers, and also for 1,900 nanometers. So for one molecule, they can absorb light at different wavelengths. And also for one wavelength, we can have different molecules that will absorb the light. So what we know about NIRS is that when we obtain a spectra, this near-infrared spectra is a specific signature of the characteristics of the matrix. It will depend on the composition of this matrix. So this is just a small talk about the NIR. When we want to use a NIR to make some prediction, to predict the composition of a matrix, of a samples, we need to have many samples. Many is not really defined, it's not really precise, but we need data. And usually we use two data sets, one about calibration. And in this calibration data set, we have to measure the spectra on the samples and also to measure the values that we would like to measure. For example, the lipid content, the water content, or whatever. And so we will do some modeling to establish some prediction equation with this dataset calibration of calibration. Usually it is with PLS methods, but we can use different methods. Then we would like to test this prediction equation. So we will test it in another dataset that we could call the validation dataset. In this one, we need to have the spectra also and to have the measure also. What we do is to apply the prediction equation on this spectra to obtain predicted values. And then we will compare the measure values, the real values, and the predicted values. And if the correlation is good, so the quality of the equation will be good. So then what we will do is on other data sets, on other samples, we will only measure the spectra and apply this method. And so we will obtain an estimation of the values. So this is the principle how we do some prediction with NIR. So today the question is to predict the carcass composition with NIRS. So the question could be how to make some spectra that will represent the whole carcass. So I propose 4 answers. One could be to make some spectra, to acquire spectra on the ham of live animals, for example, on the ear of live animals, why not? And also to measure the spectra, for example, on the whole carcass, but we have to make different measures at different points, maybe, to have an information about the whole carcass. Or another way could be to take the carcass to ground, to mill the carcass, and so to have a sample that will be representative of the carcass, and to make some measure of spectra on the top of this. So what do you think? What do you think is the best one? Actually, in the PIGWEB protocol, in our work package, sorry, we did the four measures, but the best one and with the best quality is the last one. And so I will present you some results about this part. So to acquire spectra, we need a spectrometer. We use a lab spec AST that is able to measure the wavelength between 350 nanometers to 2,500 nanometers. And for this one, we have different contact probe. So one is this contact probe with a diameter of around 3 centimeters. And we can make some measure on the meat, for example, to determine the meat quality. And the other one here, what you can see in the middle is some carcass that was ground into powder and lyophilized. And after we make some measure on the top of the samples. So here, and we measure different replicates, so sometimes 4 or 6 replicates, to really try to have an overview, a representative view of the samples. So what you have to keep in mind is that NIRS cannot scan the whole carcass, because after in the workshop you will have a talk about other techniques that can scan, have an overview of the whole carcass. With NIRS, we only have a small location for the spectra acquisition. So the challenge is really to know how to acquire spectra and to have some representative part of the carcass. But by grounding the samples, we have a representative part of the carcass. So when you acquire some spectra, here you can find some spectra. We have removed the first wavelengths and the last wavelengths because we have noise. And here we have one curve is a spectra. Here is the wavelength and the intensity of the wavelengths for each replicate. And we make some replicate to have a representative view of the samples. So here we average the data for each samples. And after, we have to make some mathematical treatments of the signal, because otherwise we will have some noise. So we will first correct for the baseline, because you can see here we have a baseline that makes a base. And we also make some normalization to, so the aim is to reduce artifacts and to improve the comparability of the spectra. So you always have to make some mathematical treatments. So now we have the data, and to have a look of the data, usually we make a PCA, so principal component analysis, so we can remove outliers, and we do it also when we have all the replicates to remove the outliers also. And what we would like after is to separate our dataset into the calibration dataset and the validation datasets. And here what we did is to use the K-means clustering methods, and we wanted to have 2/3 of the samples for the calibration datasets and 1/3 of the sample for the validation datasets. In this protocol, we only have 48 animals, so it's not a big quantity. And so we prefer to have more samples in the calibration dataset. And what is important when you want to separate your data into the dataset and the calibration dataset, and validation dataset, is really to avoid to have a bias. Because, for example, if we select this sample for the calibration datasets, and we develop a prediction equation, after it would be difficult to apply it on other samples. So it's better to really have a good representation of the samples in both calibration dataset and also validation dataset. So now we can think about what influences the quality of the prediction. So do you think it is the nature of the matrix on which we will acquire the spectra? Do you think it's the nature of the molecules that we would like to predict? Do you think it's the model of the NIRS spectrometer? Or do you think for a NIRS spectrometer model, it is the device? Is it the device? Actually, it is the fourth again. So for example, I was working with a liver before, and now I'm working with carcass, and I would like to predict the lipid contents in both matrices. In the equation of prediction cannot be the same. We have to adapt the prediction equation to the matrix, the samples that we use. If we want to predict water content or lipid content, you also have to develop on the same matrix. You also have to develop specific equations. I will show you some results just on the next slide. And we know also that the NIRS spectrometer will influence the equation. And we also know that the device itself will modify the spectra. And so we have to modify the equation of prediction, but it's possible feasible to adapt this method. If you develop a prediction model with one spectrometer, it is possible to adapt it for another spectrometer. So to illustrate the part of the nature of molecules, the effect of this, here I present you the results that we obtained in the PIGWEB experimental trial. So we only have 48 samples, so it's a very small amount, quantity of pigs. And to estimate, so we would like to predict the water concentration, the lipid concentration, the protein concentration, and also the ash concentration. And for this, we did all the first steps. And after, we compared the real values and the predicted values. And here you can find the R squared that will represent the quality of the prediction. So you can see that for water concentration and for lipid concentration, the R squared was up to 0.9. So it was good quality, because we only have 48 samples, so it's more difficult to have some good results. For protein concentration, the R squared was intermediate. Okay, I just realized that I am long. Okay, so we have then. So what we can conclude is that the quality of the prediction will depend on the molecule, but also we know that for ash, the results are not so good, but it was expected. So for NIRS, if we conclude about NIRS advantages and disadvantages, we have to keep in mind that there are factors of variation. We know that the sample preparation, the condition of acquisition, the humidity will modify the values. But what we can conclude is that these characteristics is that NIRS is really easy to use. It is really quick to make some acquisition. It can be a non-destructive matrix. Here I show you some results on the ground carcass, but we can also measure directly on the meat, for example. It is cheap. First, you have to buy the spectrometer, but after it is cheap to do. What is really interesting and good is that we can go to different places with the spectrometer and make some measure directly on the field. And we can predict some quantitative or qualitative data. But what you have to keep in mind is that we have to adapt the prediction equation for each matrix sample and also for each parameter. And also we have to keep in mind that little by little, we have to update the prediction equation, adding some samples in the data calibration. So the question was, is NIRS a good device to estimate carcass composition on pigs? And actually we see some good results, but to improve the result, we will have to improve the number of animals also. So the challenge now will be to measure NIRS spectra directly on the carcass. So we have to choose a good place to measure the spectra. And I just wanted to know that it is not a new method. On PubMed, we can find 16 publication with the word NIRS and carcass in the title or abstract. And it is interesting also to use meat to estimate meat quality and to estimate the composition of fatty acid, for example, in ham. So just to conclude, NIR spectroscopy is really a promising tool to estimate carcass composition. And I would like to thank you, everybody. And I'm sorry because I was long.
Thank you, Cecile. We can take one question directly, and otherwise, please indicate your questions in the chat. Is there anybody who has a question now? Don't see any hands. Otherwise, for the sake of time, we'll combine most of the questions later on.
Sure.
Okay, thank you, Cecile,
You're welcome.
for this presentation. So the next presenter is Örs Petneházy, who will be talking about special anesthesia for imaging. Örs has been working at the Veterinary University in Budapest between 1999 and 2005. And he started working at Medicopus and the predecessors of Medicopus since 2005 as a veterinarian. His work focuses on the cross-sectional anatomy of different domestic animals, farm animals and pet animals, and the veterinary care in human preclinical studies and in animal sciences. He created cross-sectional CT and NMRI anatomical atlases of the pig, turkey, dog, and cat. And he's the responsible veterinarian in cardiovascular human preclinical experiments that he does, for which he does the anesthesia and he takes care of the surgery of the animals. And he will talk about this anesthesia to be used for imaging. Örs, the screen is yours.
Cool. Thank you so much, and I welcome you all to this meeting. So for the beginning, I would like to highlight that it's only a very, very little part of the special anesthesia, because anesthesia by itself could last for maybe, I don't know how many hundreds of hours of workshops. So specifically talking about the anesthesia, what we are using in our institute, we are using most of the time inhalation anesthesia. So that's very important, because I think most of you have heard about total intravenous anesthesia. That's a different story. We don't use it because for long-term anesthesia and for all the imaging responsibilities, we have chosen for 25 years the inhalation anesthesia, because it's highly controllable, it's easy to use, and therefore we're using it most of the time. And since it's used normally during surgeries, that's why we're using inhalation anesthesia. So why we need special anesthesia for the pigs during imaging studies? Because we need to have the animals in total immobility. It's very important, because during a CT scan, you can't evaluate CT scans when the animal has been moving during the anesthesia. For example, if you're working on a thorax, if the thorax is moving, you will have blurry size, which is hard to evaluate. And if it's about MRI, so for example, cardiac imaging, then it's really hard to have a nice clear image of the heart when there are breath movements. So that's why it's very important that we have a controlled breathing and a controlled anesthesia during the whole procedure. The main goal during the anesthesia that it should be safe for the animal, it should be safe also for the person who is working on it. And because when you don't have a controlled anesthesia during the imaging, then when the animal is moving during an MRI, then you have to restart the procedure again and again, which means prolonged anesthesia time, which is a risk for the animal, which is also risk for the budget of the project also. So you have to keep in mind many different aspects why anesthesia is needed. And I already talked about the special problems. So for example, during a cardiac MRI or a thoracic CT, you should have breath holding and breath controlling possibilities, which is very important. Or if you're doing special imaging on the lung, for example, if you have, for example, infection studies, So what Tamás did in his PhD studies for atrophic rhinitis, or we have already different inflammation studies on the lungs, then the lung has to be in a special inflated state, because this is how we can evaluate the different parts of the respiratory system. So the main step for the anesthesia is first you have a good preparation room, a separated preparation room where we can have calm animals, and we can transport the animals 2-3 days. Some books are saying, and some special IACUC protocols are saying, you have to transport the animals at least 5 days prior to the anesthesia, that the animals are used to the environment where they have been transported. And here is where the first induction is made in a really calm, and controlled environment where the humidity and the temperature is controlled. Then after the induction, where the animals already received the first medicaments for being a really calm and sedated state, we can transport to the next stage, where we can start with the mask inhalation. And after when we deepen the anesthesia, we can do the endotracheal intubation, and then we can start securing the intravenous access, whether the ear or in the many, many different places where we already made some studies. So how many intravenous access site you can give secure in a pig. Then we do the intravenous cannulation, thermal support, cardiac monitoring, and respiratory monitoring during the whole anesthesia is very important, because this is how we can check the control rate, the heart rate, the blood pressure, and the respiratory rates. And it's very important by the pig. We have never experienced it, but in each anesthesia protocols for the pig, malignant hyperthermia, it's a very important control situation. So if there is malignant hyperthermia, you should be able to immediately act for it, because it's a life-threatening situation. If, for example, it is about a very special cardiac MRI studies, then you should be able to perform neuromuscular blockade to be able to stop the breathing, and a special moment when, for example, you are performing a cardiac MRI. If you don't have a very, very special cardiac MRI, which can synchronize the cycles with the breathing, we have it, but most of the time we rely on neuromuscular blockade, because that's the most reliable method. And of course, after the post-imaging and anesthesia period, the monitoring is really important during the awakening phase, that the animal is being awakened in a controlled environment, because no stress reaction or whatever is happening during the awakening. So before you start an anesthesia, you should do a pre-medication safety checklist. So since I do some flying with airplanes, this is what we call the pre-flight checklist. It's very important, because this is how we can perform a safety procedure. So for the first where it begins with the animal. It means that you should check the weight, the correct weight of the animals, because this is needed for the correct dosage of all the drugs what you're using during the anesthesia. You should verify that the animal has been fastened at least 6 hours. So the water shouldn't be withdrawn before the anesthesia, because then you might have a dehydrated animal, so you should leave the water there. It's not a problem. Only maybe hour before the anesthesia you should draw the water. If it's an MRI study, everybody should be awarded that no metals allowed in the MRI room, so not in the animal. We have performed many MRI studies where the animals came from an uncontrolled farm, and the pigs have been eating some metal parts outside in the farm, and they are causing a really big distortion in the magnetic field. So we had to repeat it, the MRI study on the next day after the metal has been moved out from the intestinal tract. All the cables and everything, and all the patches has to be sure contacts. that you can do the ECG correctly, the breath holding and everything. And you should make a technical checklist for your whole equipment, the gas supply, the anesthesia machines, and all parts that are available and working for the anesthesia. So for the pre-medication, the pre-medication is performed in a controlled environment. This is how we perform the pre-medication on a small piglet. You should see that the animals don't really like the pre-medication. That's kind of normal. But this is why we're using a long cable between the syringe and between the needle. We are using the butterfly syringe that you can just follow the movements of the animals so that you don't break your needle, you don't do any harm on the animal. For the pre-medication, I give you a lot of a list in the links below. So I don't give you an exact receipt what kind of pre-medication you should use. It depends on the study you are performing. We are always asking our partners what kind of medication they would like, because it depends on the study, what they want to, whether the medication shouldn't have more cardiovascular effect, it should have more neuromuscular blockade effect, whatever. It depends on always the study, what is performed. Then this coming the masking phase. So as you see, we are covering the animal with blankets that we are securing that the animal temperature is held at the constant rate. We are using non-rebreathing circuit, so for the monitoring, so for the masking effect, and then we are using a high flow of oxygen and a high flow of the isoflurane for some minutes. Then the animal would come to a resting state where you can do the endotracheal intubation. Before the endotracheal intubation, the assistant or the person who is helping you is securing the endotracheal tube with the gauze, and then the endotracheal tube is prepared using some lubricants that the larynx is not destroyed, or there is no harm is made on the larynx during the intubation. And then we are using lidocaine spray for the larynx, and we are performing a dorsal recumbency intubation. You can do it also in a sternal position. It depends on what position you like. Intubation in a pig, it's a very easy protocol. So first, I'm using a laryngoscope. I'm lifting the root of the tongue, and then I'm opening and I'm securing the epiglottis with using longer forceps, and then I'm introducing the laryngeal, the endotracheal tube. You have to check whether you are inside the trachea and not in the esophagus. So this is what we will perform at the end. And when you inflate the endotracheal tube, you should do it carefully. You shouldn't exert too much pressure, because you can harm the larynx if you over inflate the cuff on the endotracheal tube. So this is what you will see. I'm listening whether the endotracheal tube is really inside of the trachea. This is why my assistant is pushing the tracheal cage, that I can listen, that I really have the noise coming from the endotracheal tube. Then after that, we are securing with the gaze to the nose of the animal, the endotracheal tube. Then we are connecting to the rebreathing system of the anesthesia machine. And as you will see, now we are lowering the flow, because now we're using the rebreathing cycle. We are lowering the flow up to 20 milliliters per kilogram per minute. And then we are using the so-called volume support breathing system, because it means that for imaging, the animal is having its own breathing, and we are only supporting the breathing. We are not using pressure-controlled respiration or whatever special stuff. We are using it only for surgical procedures. We are securing that the intravenous cannulation is working properly. There are no blood clots. And then we are connecting the fluid therapy to the intravenous cannula. And then we are performing the imaging. During the imaging and during the anesthesia, you should have a monitoring form where in every 5 or 10 minutes, you are making the notes about the blood pressure, about the breath of the animal, about the heart rate, about the different problems what you could have, because this is what you can evaluate after the anesthesia, whether you performed a good anesthesia or not. For emergency situation, you have to be secure that you are prepared for any kind of emergency. In the case of malignant hyperthermia, you can use the dantrolene, or there is for bradycardia, you can use atropine, epinephrine, or lidocaine if you have any kind of arrhythmias. It is really important that you have this on site and then you are prepared, because you have maybe 10 seconds to react or maybe less time. So you don't have time just to ask for where is this syringe, where is that syringe. All the syringe have to be labeled, and all the drugs, the different drugs for emergency have to be on site. So here I made a list for you where you can have special anesthesia protocols and everything according to different university needs and IACUC protocols. And if you have any questions, that you can contact us, and we can plan special anesthesia plans for you if you need any kind of help. Thank you so much for your kind attendance, and I'll give you the word back to Jaap.
Okay, thank you. Thank you, Örs. I don't see any questions yet. Yes, I see a hand raised. Leah, please, just we take one question and then other questions later on. Leah, go ahead. Unmute yourself. Hello, Leah. I don't see her anymore.
I'm sorry.
Yes.
There was a mistake by myself. I wanted
Okay.
to check the chat because I thought maybe there was an interesting question. Sorry.
No problem. No problem.
But I like I understood.
Yeah, go ahead.
Probably most of it. So I'm new to the topic.
Okay.
So,
great.
Yes.
So if you have questions, you can ask them later on. Great.
Okay.
Thank you.
Thank you so much.
Yeah. Thank you for this. So now we move on to the third presentation by Claudia Kasper from Agroscope in Switzerland, and the use of DXA in pig production. Claudia has been a researcher at Agroscope for many, many years in the animal genophenomics group. She has numerous years of experience in applying dual energy X-ray absorptiometry, DXA, to determine body and carcass composition in pigs, including fattening pigs, sows, and piglets. She has contributed to the development of equations translating DXA measurements into chemical determined reference values. And Claudia uses DXA to estimate muscle mass in live pigs and carcasses to quantify nitrogen use efficiency in genetic studies. In September, along with colleagues at Agroscope and other colleagues, Agroscope was hosting a collaborative experiment as part of the PIGWEB project, using multiple non-invasive techniques, some of which you will see today, including 2 DXA devices located at Agroscope, one installed in the barn for scanning live animals and one in the slaughterhouse, which is fully compliant with cooling chain and hygiene requirements. Claudia, it's all yours.
Okay. I hope my screen is visible now.
Yes, it is. Sorry.
Yes. Okay. Thank you. Thank you, Jaap, for the nice introduction. Good morning to everyone, and I hope I can convince you that DXA is a good tool in pig production, especially when it comes to body composition in pigs. So by body composition, I mean the weight of different tissues, such as bone. In chemical terms, this would be ash, or fat, and in chemical terms, lipid, or muscle tissue, which is protein or nitrogen. And what kind of research is it needed for? So in general, nutrition, for example, bone mineralization in sows following different diets containing phosphorus and calcium, for example. My personal highlight is protein and phosphorus efficiency determination. You can also check for compensatory growth, or how an animal can cope with environmental stresses. And in the end, of course, you can also use it for carcass grading. So as Cecile has already talked about, what is needed is a reference method. So what we use or have used previously techniques that we really trust, that we consider as true values. For example, chemical analyses. But they are usually destructive, so they need you have to slaughter the animal and destroy the carcass, so you cannot consume it anymore. So it's really a waste. And why we want non-destructive methods, of course, is that we could assess the body composition on live animals even. Then we can still use them for breeding, or we can use it on slaughtered pigs and keep the carcass in the food chain. So the traditional reference methods are, as I already said, chemical analysis. This requires the carcass to be ground up and to be analyzed with wet chemistry methods, or also dissection by knife. So you need highly skilled personnel, and it's quite time consuming. Unfortunately, I did not dare to provide you with videos, but I have some photos of how we do the carcass grinding for chemical analysis. So here, Esther, the PhD student, was transporting the frozen carcass cuts in to the machine that grinds them up. So this is the machine. This is the bottom of the machine where the minced meat comes out. So this is half of a carcass of a fattening pig ground up, and it looks like minced meat, but of course there are also the bones in it. And in order to obtain representative sample, we mix it for a few minutes. To have a really homogenized product, and then we can take a sample out of it. So this is what we do, but you see it's really time consuming. Similarly, this photo shows a team of butchers during our PIGWEB trial who really cut all the tissue types from each other by knife. So this needs around 10 hours per carcass per butcher. So this is really something not so easy. Of course, also besides the non-destructive part, we want something cheap and fast if possible. So the reference methods, as you have seen, are laborious, they are time consuming, they are expensive. They can usually be only done on small sample sizes, so the throughput is really limited, and they are destructive. Here comes dual energy X-ray absorptiometry. In brief, it's DXA. And it's quite a simple setup. So here you see the photo of the DXA table. Usually where you see the hole there, it's covered with some wood panel to put the body on. And below the table, there is a part that emits X-rays. So the orange arrow shows the way of the X-rays. They go from below the table to the upper part. And the green arrows show the movement of the DXA arm. So to really scan to the left and to the right, and then it moves along the body from head to toe. And this DXA comes from, is used in human medicine, also for body composition determination, especially bone density, osteoporosis. And it emits X-ray beams with 2 different wavelengths. And these are emitted under the table. They penetrate the body, and then they are captured on top in this detector. And different types of tissues attenuate the beams in different ways, depending on their density. For example, bone is, of course, very different, has a very different density than lipid. And by this, it's possible to distinguish bone, lean, and fat. Here is a closer look at the sender part. So this is the part where the 2 X-ray types are emitted. And on the right, there is the detector, which captures the attenuated beams. So what you get in the end is a 2D projection of the body. But fortunately, the technique has advanced, and instead of having one beam that directly horizontally, vertically penetrates the body, there is now a technique that with a fan beam, so that's a spread out beam that can, up to a certain degree, avoid shadows of dense tissues that obstruct other tissues, for example.
The scan duration is 8, but rather 12 minutes, depending on the size of the animal. Of course, for smaller animals, it's finished sooner than for larger animals. And you can use it for scanning live pigs or carcasses or even parts of carcasses. And the results are an image that you can easily process afterwards, and then you get a simple result table indicating the weights of different tissues in the total body or in the regions, in the body regions that you can define during the image treatment step. Here are some photos of how we do scans in live pigs. As Örs has already mentioned, also in our institution, we usually use insufluran inhalation anesthesia. Here is the pig with the mask. This is a piglet. We have different sizes of masks. It works for all sizes quite well. For the PIGWEB trial, we wanted the pigs to sleep a bit longer, so our veterinarian injected them with a cocktail of several anesthetics. And so it can sleep without a mask. And in the end, what you get is this image. This is one of a scan with the mask. So in blue on top, you see the mask, but this can be manually removed from the image in the image processing step. For carcasses, we can do that, as Jaap has already said, while observing sanitary rules. We can cover it and we can have it in a cool place. And this is the outcome of the carcass scan. Of course, then the orientation of the body is a bit different. It lies on the side instead of in a prone position. On the on the left, you have the classical X-ray view, visualizing the bone. And in the middle, you see different tissues. Like in green, it's the green is the lean tissue and red is the fat tissue. And on the right, you see the resulting table. Of course, it's a bad photo, so it's not really visible, but you can get the idea of what you get in the end. We also scanned single cuts, a bit separated. You can also manually separate them then in the image treatment step. And this is a photo from yesterday where a colleague is taking scans from feet to see whether it corresponds to their composition correspond to the total body composition. So of course, what we want to know is how well can we trust the results of the DXA, the values that come out of DXA. This is a machine calibrated for humans, so of course we really need to know how good is it. And this is why we did a study comparing chemical values and DXA results. And yeah, just to remind about accuracy and precision, what we want in the end is on the bottom right, we want values that have high accuracy and precision. So they are right on the spot of the chemical values. But what we can hope to get with DXA is on the right top. We hope that the values are not very much spread, but they usually will not be exactly the same absolute values. And this is why you use regression equations to convert DXA to chemical values. And we did this study and looked at our precision, and we found that for crude protein, nitrogen, and lipid, this looks very nice. The correlation is good. On the y-axis, you see the chemical values, and on the x-axis, the results given by DXA. And I mean, it's clearly visible that the cloud of points is quite small. It has a small spread. As Cecile has already mentioned, we run into the same problems for ash or bone tissue and the bone minerals. So this is a bit harder to predict. We have more spread. So if you're interested in this study, I have provided the QR code here. So what can you expect in the end? You can expect medium to high precision for body composition with a low bias with an error of 4 to 13%, 4 for protein, 13 for bone. The good thing about DXA is also it has really low maintenance costs, and to operate it, you need only medium technical skills, so it's not difficult to learn. The work security is quite high because it emits quite low radiation. And after you have acquired the image, the treatment of the image is really fast. It can be, in the case of carcass, a few minutes, 2 minutes, or when you have a mask to be removed, of course, then it takes a bit longer, but usually in 5 minutes you are done. Unfortunately, you cannot do volumes of certain body parts or virtual dissection in slices as with CT or NMRI, and you have this more or less 2D projection, and the duration is quite long, so it's almost 15 minutes. And as a take-home message, I would like to give you some applications in research in our institution, where we use DXA to look at bone mineralization in sows, or determine the heritability of nitrogen and phosphorus efficiency, or predict the body composition in cattle from scans of cuts. Of course, a whole cow has no place on the DXA table. This is too large. And in livestock industry, in Australia, it's commonly used for lamb composition prediction in abattoirs and also for breeding decisions. Thank you very much for your attention.
Thank you. Thank you very much, Claudia, for this presentation. I suggest that we move on right away to the next presentation. And again, please put your questions in the chat or remind them and then ask them later on to Claudia and the other speakers, of course. Thank you, Claudia, again. So the next presenter is Maria Fonti Furnols from IRTA in Spain, Catalonia. Maria is a researcher at IRTA, the Institute of Agri-Food Research and Technology. Since 2000, she's been working there. She belongs to the Food Quality and Technology program and works mainly on carcass and meat quality. She has a great deal of experience in using CT scans for estimating, for characterizing body and carcass composition, and in the sensory analysis and consumer studies for meat quality determination. IRTA is the reference center in Spain for pig carcass grading, and Maria is the national expert in pig classification. She has participated in classification trials in Spain and abroad and evaluates EU protocols for the authorization of pig carcass classification devices. Maria, that's up to you. Please put it in full screen.
Now, can you hear me?
I can hear you, but it's not in full screen.
Can you see the?
Yep. If you do that, great.
The full screen, right?
Yeah, thanks. Yep.
Okay. Well, I'm going to talk. Some things are about the use of computer tomography. Some things are similar to those that has explained Claudia with DXA. And first of all, I like this image because it shows what we can see with the computer tomography. In fact, it's an X-ray, so we can see the interior part of the animals, of the carcasses, etc. And I like to include Superman because he's flying, so we can have the city in a trailer, so we can move everywhere to use this technology. What it is, this technology? It is an X-ray technology. There is an emission of X-rays in the gantry, which is this round piece here, and they go through the body, and there is a detector in the other side that is moving. And from all this, it collects the attenuation values, and with these values, using a reconstruction algorithm, it produces an image. This image, even we see it in 2 dimensions, it is a 3-dimensional. So instead of pixels, we talk about voxels, which is a pixels in 3D. The images are in a gray scale, and it goes from low density in dark to high. This is low density in dark to high density in lighter part. So we can have this density is evaluated in Hounsfield's value. The 0 is the water, a little bit higher of 0, from 0 to 120, 140 is the lean. From -1 to 140, 145 is the fat. The air is -1000, and the bone is very variable because the density of the bones is very variable. So what we can see if we scan a pig is an image like that. So we can have axial image of all the body of the pig. I'm going to do it quick. So we can see the head, the shoulder, the central part of the pig with the loin, the tender loin and the kidneys, and at the end, the ham. Then also, we can also scan carcasses. In this case, we don't see all the part of the viscera, but we see the ham part, the loin, and also the shoulder. So then, with all these images, what we can do with all these images, I have said that it is a 3D image. Even we see into this, so we can see taxially, as we have seen. We also can see sagittal, an image, or a coronal image of the animal. And from each of them, we can take linear measurements, or we can take areas. So we can get a lot of information, because from all the images, we can take whatever we are interested in, depending on the objective of the project. So also, since it is a 3D image, we can construct the animal in 3D, and we can move the focus to see only the bones, for instance, like that, or we can also cut it, do a virtual cut at different places. So also, from all the images, we have the voxels. For each voxel, we have a Hounsfield value, and we can calculate the volume associated to each Hounsfield value. So we get an histogram like this one. Here is for a carcass in red, another one in blue, and another one in green. In the negative part is the fat. In the positive part is the lean. So which is the leanest carcass here? Is the green one, which is the fattest one, the red one. From this and this part here are the bones. From the images, we can work directly using segmentation techniques, or we can work with this histogram. And in all of them, we need to do some statistics, for instance, multiple regression or PLS, in order to get the parameter that we are interested in. Which is the advantage of this technology, is a non-destructive technology. So we can scan the same animal at different moments during growth. So we can do serial scanner, avoiding the serial slaughtering. So it's a good way to reduce the number of animals that we apply, that we use in one project. We also can have the measurements or area of fat thickness at different moments during growth. So we can see the evolution of the loin area, for instance, depending on the genetics, in this case, Pietrain and Landres and Duroc. Also, we can see the evolution of the fat depending on the sex. For instance, the castrate, which are the fattest one, the females and the entire males. And here we include the immunocastrated that are like entire males up to the second vaccine, and then they deposit more fat and they increase. Also, we can see the effect of the diet. For instance, we can see an animal, the growth of the fat area in an animal that it's fed at libitum, another one that is fed at libitum at the beginning and with restriction in the last part, or another one with restriction at the beginning and at libitum at the end, where we can see this compensatory effect. Also, from the images in some studies, it could be of interest to distinguish between the different types of fat. For instance, we can evaluate the subcutaneous fat, we can evaluate the first fat or the visceral fat. Depending on the objective of the project, we can do one thing or another one. Also, as Claudia has said, the bones are very important. We can scan the bones alone or look at the bones in the whole animal. And from the bones, we can see the length, we can see the area or the thickness of the cortical part of the bones. And as I have said before, if this is the histogram, this part, this last part in the right part are the bones. But it seems that all is the same. But if we look in detail, we can distinguish between these animals. And this is the bones, the part of the bones with low density and in light blue, and this is the part of the bones with medium and high density in dark blue. So if we have studies in which we add, for instance, phytase in the diet, we can see the effect of the phytase. So as the phytase increase, it increased the proportion of more dense bones. So there is an important effect in the density of the bones depending on the feeding. Also, it can be translated to a value of density applying an equation. And well, just to finish, we also can use it, of course, in carcass and cuts to determine the carcass and cuts composition. And we will see it in the last talk today that also the CT can be used as a reference for calibration of online devices of pig carcass classification at a slaughter plant. And that's all. Thank you.
Sorry, I had to find my button to unmute and my screen. Thank you, Maria. I will have some questions later on, but I'll wait for the discussion if nobody else has questions. Thank you for this. So again, keep your questions in mind or write them in the chat, so we will treat them at the end of all the presentations. Thank you, Maria, again. So the next talk is by Tamás Donkó from Medicopus in Hungary on NMRI examinations of pig carcasses. Tamás has been a researcher at Medicopus since 2007, and he's currently the head of the research unit and honorary associate professor. His research unit conducts preclinical research as well as livestock-related studies using imaging diagnostic methods, as well as CT scanning and NMRI imaging. His main research focuses on the adaptation and evaluation of imaging techniques for agriculture purposes and their practical application. He regularly supervises BSc, MSc, and PhD students. Tamás, the screen is yours for your presentation about MRI.
Thank you. Can you see my shared screen?
It works.
Okay.
Yep. Great.
Okay. Good morning. I would like to share you our work on the PIGWEB project, focusing on the use of NMRI in carcass evaluation. First, let's clear something about NMRI, because most of people just call it MRI because the word nuclear tends to scare people, but don't worry, there is zero radiation in the magnet. Here we're just talking about the nucleus, so like an atoms of the examined object, in our case carcasses. So from our point of view, the carcasses or living animals are like basically a big bag of water. I mean, they are full of hydrogen protons. Normally, these protons are spinning around like crazy in every direction. But what if you slide into a giant magnet? They all line up like soldiers. You can see here, and their behaviors is like small magnets. That's why this phenomena. And then when we get an image, we have to disturb them. We hit them with a radio frequency pulse, basically a targeted radio wave. This knocks the protons off balance. maybe you know those loud, annoying sounds the machine. Those are the giant coils. They are changing the magnetic field in tiny increments, so the computer knows exactly where in your body each signal is coming from. No noise, no CD map. So that's why the noise exists during the examination. And when we turn these paths off, they return the normal place. As they do, they send the tiny radio signal back to us, what the coils collect. I will explain the coils later on. The raw data collected isn't an image yet. It is a mass of frequencies called K-space. So a complex mathematical process called the Fourier transform is used to convert those frequencies into the high-resolution grayscale image like X-ray, DXA, or CT can produce. I close this physics part for now. We will get back to the technical side later, especially the MRI sequence. But first, I want to clarify why we were even doing these MRI scans in the PIGWEB project. As part of the PIGWEB project, we had the opportunity to conduct a collaborative experiment, as we mentioned before, and we used the equipment on the same carcasses. We created the name Haribo by combining the key letters of the study full title. And you can see Cecile actually brought us a whole pack of Haribo gummy bears here to stay focused. And within the Haribo experiment, one of our challenges was accurately determine the mass of the different tissues in the carcasses using various instruments. To do that effectively, we turned to a very specific MRI technique. Let me show how it works in practice. Our Swiss colleagues labeled and packed the right carcasses carefully and sent them to us in a freezer tag. Then we split the truck cargo area into two sections. The inner part was kept at -18 Celsius, while the outer part was at 6 to 8 Celsius plus. The carcasses waited in the freezer section until it was time to move them to outer section for controlled towing. It is, in this photo, you can see my colleagues. They were controlling and checking the core temperature of the carcasses. And why we do this? Because, as I explained earlier, it's impossible to get an MRI signal from protons trapped in ice. They have to be in liquid form. As you see the graph, the carcasses tow safely after about 2 or 3 days. So we scan them on the 4th day of the towing period. We transported the towed carcasses to the MRI machine, along with the feet, chunks, and the head. We placed these smaller parts on the table first, making sure that didn't touch each other on the main carcass, or the main carcass, yes. It is very important. It makes easier to segment the different parts after that from the images. To keep them separated, we used these pieces of foam padding. I tell you the fact now that the magnetic field is a standard 3 Tesla NMRI is about 60,000 times stronger than Earth's magnetic field. This is why, as Örs mentioned before, definitely don't want to bring your keys into the room. Okay, it's a serious thing, safety first. So after that, we covered everything with plastic foil. And okay. And we placed the coils, into the carcass. But what are these coils and why do we need them? In the simplest terms, coils are antennas. Remember how I said the protons send back a tiny radio signal. Well, the coil is there to catch the signal as clearly as possible. So the closer to coil to the body, the better the signal to noise ratio. So that's why we place them directly onto the carcass. It's like putting a microphone right in front of the singer instead of recording from the back of the room. So without these coils, the signal would be weak and blurry. They are the key to getting those sharp, high-resolution images we need. As I've mentioned in the study, we used the T1 weighted Dixon sequence with the setting shown here on the slide. Every examination starts with the localizers. These are quick scout images used for orientation, and then we got the right position of the carcass. The machine knows where is the carcass. We plan the Dixon measurements. For a single carcass, we perform 8 separate measurement blocks to cover the entire body accurately. The table moves automatically so that every image ranges are captured in the center of the magnet. This is crucial because the magnetic field is most homogeneous right in the center, ensuring the best image quality. The acquisition process takes about 45 or 50 minutes. So the whole process, the change of the carcasses and positioning is about an hour. Okay, let's talk about the Dixon sequence deeply. Why did we choose this, especially for measuring fat and muscle tissues? Because 2 for the price of 1. So the biggest advantage is the efficiency. Instead of running separate time-consuming T1 or T2 weighted scans, the Dixon sequence gives us everything we need in one single measurement. During that one scan, the machine captures 2 types of images, the in phase and the opposite phase. And this is based on the fact that protons in fat and water spin at slightly different speeds. So by adding or subtracting these 2 images, the computer can mathematically separate of the signals. This gives us 2 perfect sets of images, the fat-only image and the water-only. It's fast and accurate and gives us the exact volume of fat and muscle using a simple and automatic segmentation method. As you might notice, there were some intensity differences here between the 8 measurement blocks. My colleague Daniel corrected these variations, so the final images you see here already smooth and uniform. I haven't mentioned one major disadvantage of the MRI yet, because both contain very few protons, the small magnets. MRI actually not suitable for bone imaging. This is where CT has a clear advantage. While MRI is king for fat and muscle, CT is far superior for bone structure, as Maria mentioned before. Okay, I won't go into all the detailed results, but I show you this graph because this figure demonstrates the relationship between the lean meat ratio measured by CT and the results we obtained by MRI. As you can see, there is an incredibly close correlation between the two methods. This proves that our MRI protocol is just as reliable as CT for determining lean meat content of carcass. So finally, let's look the big picture. What are the real advantages and disadvantages of using MRI in this type of research? The main benefit is that is non-destructive, so the carcass remains intact. It is much more important in live animals, but we examined only carcasses in this project. It also gives us incredible soft tissue contrast and allowing both high quality qualitative use and precise quantitative data. On the other hand, there are limitations. As we discussed, it's temperature dependent, which is a specific challenge for carcass measurements. It's also, it is not a problem with living animals because the temperature is the same. It's also slower and more expensive than other techniques like CT or DXA. And unlike ultrasound, this machine is definitely not comfortable or NIRS. So we have a stable one. But I know somewhere exists mobile MRI, but it has, I think, higher cost than us. In conclusion, why NMRI comes with certain logistical technical challenges, such as temperature sensitivity and long acquisition times, it remains a powerful tool for soft tissue analysis and examine living animals, not for carcasses. And finally, I would like to thank my colleagues and our collaborative partners for this unique opportunity to examine 48 half carcasses. It has been a privilege to work together on such an exciting project. Thank you.
Thank you, Tamás. I think this is one of the most interesting studies I've seen in my life, I think, in terms of gathering people around, as you see, around a carcass, so and a live pig. So we save the questions for after the last presentation that Maria will do again. I will not introduce her because she was already talking just before Tamás's talk. Maria will be talking about pig carcass grading in Europe and classification devices. Maria.
Yes. Okay, this is a little bit different topic, but it's related. But it's more related of what it is done in slaughter plants regarding the pig carcass grading. I want to start with some introductions because I think it's important that we understand that in Europe, all the pig carcass grading is legislated. There are regulations at European level, but also regulation at national level. And why they are important? Because we do the classification at the slaughter plant, and with this classification, we can pay the producers. So economically, it's very important. But also, this classification can help to establish some price of the meat. So the calibration is of the devices that are used to determine the limit content and the way the carcasses are treated are very important, and they are regulated. And also, since there is a calibration and there is an examination of the carcasses and evaluation, we need a control of the devices that they are working correctly. And all this is legislated. So why do we need all these legislations and what we can use? Why do we need the classification? Because it allows us to compare the prices between member states and also the pig population between member states. It also allows a fair payment to the producers and it's a higher market transparency. I will not enter a lot in the legislation because it's very boring, but I think we need to talk about the three main laws, that the three main regulations that are updated now. This is the first one that defines what it is a carcass, which is the presentation of the carcasses, because this is very important because it affects the price. So if the presentation does not include the flare fat, and you include them, it affects at the price. If the presentation includes the feed and you remove them, it affects at the weight and at the price. So it's important to have an official definition and adapt the weight in case that you remove some of the pieces that should not be removed. Then it classifies the limit percentages in classes, syrup, depending on the limit content. And also it says that this content, these grading methods need to be authorized by the Commission and after a process of being statistically proven. And I will, these two legislation establish how this calibration needs to be done. It's by means of dissection trial with a minimum of 120 carcasses. Then I will enter on that. We need, of course, to record the measurements in the grading techniques that we want to calibrate. And then we need to, this 120 carcasses minimum need to be dissected, either fully dissected or with this equation of the left, or partially dissected the loin, the shoulder, the loin, the ham, and the belly. And but if we use the partial dissection, we need to find the bias in order to mimic the full dissection. So because the reference is the full dissection. All these prediction equations need to have an error lower than 2.5, calculated with full with cross-validation leave one out. And this dissection, which is time-consuming, can be replaced by CT on the condition that satisfactory comparative dissection results are provided. So as a summary, when we do a dissection trial to calibrate these devices online that we are going to use online in the slaughter plant to classify carcasses, we have the measurements from these devices, so we have the predictors. Then we need to do the dissection of the carcasses. This means by simplified dissection to fill these green boxes here, skin and subcutaneous fat together, but we can fill these green boxes here and calculate the limit content. Or with full dissection, we need to fill the green and the red boxes here. So for simplified dissection is 4 or 5 hours of one technician per carcass, one batcher per carcass, and for the full dissections it's around 10 hours for one batcher per carcass. So it's really very time consuming, but if we have the CT calibrated, we can use CT instead of the dissection. From the predictors and the limit content, we can get some relations equations because there are relations between, for instance, the fat thickness and the limit percentage, and a linear regression like this one, etc. So these relations, these equations need to be approved in Brussels, and they are published in the official journal of the European Union. And we need to have in mind that each device has its equation, and this equation is different per country if the pig population is different. This is just an introduction. Let's see which are the type of devices that we can have. We can have devices that can be classified depending on the technique. They can be vision devices, they can be reflectance, ultrasound, or electrical conductivity. Also, they can be classified depending on if they are fully manual, if they are semi-automatic, or if they are fully automatic. In this case, for instance, I am going to show you now some of these devices, not all of them, because I don't have, there are a lot, but a little bit. So I'm going to show something about the ruler, which is not a real device, it's just a ruler with some vision, semi-automatic and automatic devices, some reflectance semi-automatic device, and some ultrasound semi-automatic and automatic devices, and also an electrical conductivity automatic device. Before starting, just to see which are the devices that are approved in Europe, we can see that 55% of them are semi-automatic, 28% are automatic, and 16% are manual, mainly ruler and Agroscope. Also, if we classify depending on the technology, we can see that 45% use the reflectance, 24% the ultrasounds, 16 the vision, and ZP method 10%, 11, but this is only for the smallest water plants. So let's start with one of these devices, photometer, which is similar to the way of working is similar to the Hennessy grading probe, the Capter Grass Magre, and the Destron. This is an equipment that works with reflectance, and it is semi-automatic. Semi-automatic, it means that there is an operator that takes the measurement, and but the measurement is collected automatically. So the operator, the technician, does not need to read the measurement. It is taken automatically. In this case, it takes a fat thickness and a muscle depth at a specific level. For instance, if you see, they are counting the ribs before taking the measurement, because in this case, this is in my country, and in this case, we measure between the third and fourth last ribs. So they count the ribs. You can see that this is an invasive method because it makes a hole in the carcass, it measures, the proof enters the carcass, and when it goes out, it measures by reflectance the difference between lean and fat. It measures, and depending on the distance, it measures the distance. And these are the lean and fat are good predictors to determine the lean meat content of the whole carcass, measuring only in one specific side of the carcass. Another technology which is very similar to the photometer or the Destron is the ultrafoam. Is very similar to the previous one. Is semi-automatic, but it works with ultrasounds. In this case, since it's ultrasounds, is not invasive. You can see that here there is not a hole in the carcass. It has this advantage with respect to the other one. This is a fully automatic device, the CSV image meter. We are going to see later on the VCS 2000, which is similar. This method, this technology has a camera, and with the camera we get some images, and from the images some fat areas or muscle areas and fat thickness and muscle thickness are obtained, that are used as predictors in order to obtain the lean meat percentage of the carcasses. Another methodology similar to the previous one is the VCS 2000, also a vision system and also a fully automatic system. In this case, there are two cameras, and here it's important to have light enough to see very well the images in the carcass. Another very highly used equipment in Europe is the autofoam. Autofoam is an ultrasound technology. In this image, you can see two autofoams. This is because this belongs to a trial, and we were testing two autofoams, autofoam 1 and autofoam 3, but usually there is only one of them in the line. You can see that in the previous devices, we evaluated the half carcass. In this case, we do the evaluation in the body, in the whole body, after scalding and deharing. So it's different. In this case, also, as well as the vision system is non-invasive, non-invasive technology, and it's fully automatic, and the speed can be very, very quick. There are 16 transducers in this U-shape equipment, and it measures along 1 meter of the carcass. It can also be used from all the data obtained. You also can predict the weight of the primal cuts and also its lean meat content. Another device that is less common, as far as I know, it's only approved in 2 countries. It's the GM scan. It's a magnetic field distribution across, is a kind of use. It works with magnetic induction technology, and it's also fully automatic. You can see more or less an image here, how it is working. In this device, the weight, usually the weight of the animal, of the carcass, sorry, need to be included as a predictor, because it depends on the weight quite a lot. Another method, which is more simple and everybody can use it, is the ZP method, which is a measure of the muscle, of the fat thickness over the muscle gluteus medius, this image in the left, the minimum fat thickness over the gluteus medius, and the fat muscle depth between the vertebral canal to the end of the gluteus medius. This method can be measured in a ruler like this one in the video, or also with a calibrated ruler like this one in the image, where you get directly the limit content with the measurements. This method, of course, it's slow, and it only can be used in slaughter plants where the speed is not very high. Otherwise, it's not possible to use it. And based on these ZP measurements, there are some devices semi-automatic, like the OptiScan TP. This is based in vision, and you can see that it takes the measurements obtained with the ZP. First, you can see in the video, first it measures the fat thickness here. Now it's measuring the fat thickness, and then it measures, this is the fat thickness, and then it measures the muscle depth. You can see that it's like a ZP method, but it takes the measurement automatically. So it's a semi-automatic method. All these semi-automatic method needs an operator, and there can be an error of the operator. The fully automatic methods don't need an operator. All the devices need to be calibrated before using, of course, and there need to be some maintenance and controls. And well, just to finish, some thoughts about it, which is the best method. That depends on what you need. That depends on if you use it in a slaughter plant. That depends on the characteristics of the slaughter plant, the capacity, the speed, the space. If it's very quick, even the semi-automatic methods are difficult because they need an operator. Sometimes in a very quick slaughter plants, you have two operators, and they do one carcass, and they one carcass each in order to be able to have all. The fully automatic methods have no problem of speed usually. Also, other aspects are the failure frequency, the accuracy, the maintenance feasibility, the data management capacity of the slaughter plant, the easy or difficulty to use, the requirements of staff, or the investment and depreciation. Well, I hope that you have an idea of what it is, the carcass grading in Europe. Thank you.
Great. Thank you, Maria. Thank you for this presentation. We've seen the different technologies, machines being used in commercial operations like Maria has shown, or for research. I suggest that the presenters unmute their microphones so you can respond to questions that people have. There are a couple of questions on the chat. One is by Cornelia Metges. Given that CT and MRI give comparable values for lean and fat in carcass, what justifies the use of a much more expensive NMRI technology? Does it provide more or specific information that cannot be obtained from CT?
Yes. I answered this question shortly. We use CT for this purpose as well as Maria do that. So the NMRI much more good option if we would like to know, for example, the heart capacity, or we would like to know something about the heart infarct size, for example, what we examine regularly. But for this purpose, I think NMRI is too difficult, and as I have mentioned, we have to have lots of standardized things like temperature and positioning, and we have to avoid the high temperature and low temperature as well. So it is not so easy to handle. And after that, we have lots of post-processing work with the images as well. So basically, I can tell you the truth that the NMRI is not the first option for carcass examination. But if you would like to have, for example, spectroscopic data from different places of the muscle, you can do it. And for example, the intramuscular fat can be measured by this technique, but it is time-consuming as well. So we have other options, which is cheaper and quicker. So we can do it, but not, the NMRI is not the first option, I think.
Okay, thank you. And also in terms of mobility, the CT scan, I mean, Maria can put it on a truck, which is...
And NMRI can also be in a truck. There are some, for medical purposes, there are some.
Okay.
And the advantage mainly of MRI of CT is that you can see the bones, and it's very quick. We scan one live peak in 1 minute and not much more. So, well, that depends on the power of the device, of course.
Yeah.
But it can be really very, very quick.
Can I ask a follow-up question on this issue? Claudia said the CT, the DXA is basically two-dimensional, but can you take multiple scans, and by positioning the carcass differently, could you, by scanning the animal from different positions, could you create a 3D image by DXA?
To be honest, I doubt that. I mean, I don't know if we have the software to put to kind of puzzle the picture then together. Because I think it's very crucial how you do that exactly. So I'm not aware of any.
Okay. And that brings me to another question, basically for all three of you, because you know we know that if you scan a pig, irrespective of the system that you use, you will see tissues, you will see organs, and by anatomy, you know, especially with the NMRI and CT scan, you know what the liver looks like, you know what the heart looks like. Could we use artificial intelligence to basically identify these organs specifically just by scanning them and saying, hey, this was the heart, this was the liver? Is there, because as you say, Claudia, the data analysis, the analysis methods are complex, but is there any possibility in the future to use artificial intelligence basically to train our equipment to say, this is this organ, this is this organ? And even if you go back to Tamás, this is intramuscular fat. Could we imagine that?
For the organs, I think it already exists something at the medical.
For humans.
Yeah. Yes.
For humans, yes.
And for pigs, is that something for the future?
Yeah.
Why not?
My colleagues work for that.
Why not? Because at the end, pigs are similar to humans, or humans are similar to pigs. Make it as you want. For intramuscular fat, we try it with CT, and it's really, really difficult to measure because we can have 1 pixel or 1 voxel, but with intramuscular fat, we don't have a full voxel of fat. So it's all mixture voxels. So it's really difficult to identify. Then you need to do it through regression equations. And at the end, we did it with I say acceptable prediction, but it's not the best technique. Maybe MRI is better for that, but CT is not the best one for intramuscular fat.
Okay. There was another
I would
question.
like to
Yeah,
add
go ahead. Yeah, go ahead.
for
Go ahead.
DXA, if there's still
Yeah,
time.
sure. Go ahead.
For DXA, we have the disadvantage that we have no access to the raw data. So this is an opaque box. And I think this makes it really difficult to train something on it because we don't see the real data.
Yeah. Okay. Yeah, no, makes sense.
Mm-hmm.
There's another question on this chat from Baguma Salomon. I don't know what is the first name or last name, but for a request to partner with the South, because this was an EU product, and to what extent can partnerships be established? Because this is really about PIGWEB was about the research infrastructures and to share them, to make them open. So to what extent are these facilities open? They can or accessible financially, partnerships-wise? How do you see this in the future to come? Can we ship carcasses to Hungary, or can we move the truck from IRTA to Africa if needed? I mean, not talking about cost, but is it?
It's possible. In fact, this is one advantage of having the device inside the truck. In fact, for the PIGWEB study, I did not focus on PIGWEB results, but we moved the truck to Agroscope, so we can use all the devices at the same time. We have been in Serbia, we have been in Italy. Now the city is in Italy at that moment, so I think it's good. It's easier. It's within Europe, because the paperwork is much easier than it is of Europe, of course, but all is possible. Yes.
Claudia, Tamás, how accessible is?
We are open the next grant, and we are open for cooperation with different research organization in Europe or outside in Europe. Basically, we try to find a good cooperation with others. So the best quality of the publications comes from this type of projects, I think.
Okay. So if you have a question, please raise your hand so we can see it. I have a question for Cecile. Because the relation with chemistry, so not talking about lean or fat, but with protein and lipid is sometimes important, quite often important for nutritionists. And you said that the method the spectrometer and the NIRS can be used, and is best used for homogeneous samples, and for tissues it's more complicated because it's composed of protein and lipid and water and different. To what extent do you see a possibility that intact tissue samples can be used to estimate chemical body composition, and not just on one location, but with the cheap device like the ones you illustrated, you can take easily for research multiple scans at multiple regions. And how can that be representative for the overall body composition of the animal, the chemical body composition of the animal? Is there any future to use that technology?
Thank you, Jaap. So I don't know if I have the exact answer, but I can try to add some information. NIRS is really good to predict the composition of what we measure. Before I was working in liver, in fatty liver in ducks, and I wanted to predict the lipid composition of the fatty liver. And I measure spectra on the fatty liver, and the correlation is really good. So we can work with a tissue. It's really not a problem to work with a tissue. But what we wanted in this work package was to estimate more the carcass composition. And so my question was where I have to measure. So maybe we had to do it also in the same place exactly where we measure usually the carcass on the slaughterhouse, but we didn't do it. So I don't have the values. But for example, we take some spectra on the top of the meat of loin, and we wanted also to predict the carcass composition. And for this, it was not perfect, but we have some interesting results. The correlation was for protein, it was around 0.6. And so measuring just the meat, of the loin, we wanted to predict the carcass. So it's normal that we don't have this so good correlation, because we are not estimating if we estimated the protein or the lipid of the meat and we measure on the meat, I think we have very good results, even if we have small amount of samples. So I don't know if it would be the next advice. It is less expensive, I think, than the other one. I know that there are other studies for example, to study the fatty acids, exactly the composition of fatty acids in meat or ham, for example, in Iberian pigs. It's possible, but I'm not sure that we will replace what is done already.
Yeah, because all the techniques that you presented are, I mean, I will say the gold standard, just slaughter the animal and dissect it completely and analyze it completely chemically. It is very easy. I mean, in principle, but it is very complex and time consuming to carry out. And so that's why we have these imaging analysis techniques to have some other indicator. But to what extent can we go from tissue to chemistry? Is that a big step? Is that a small step? Is that how suitable are these different techniques to say, okay, it's not just protein and lipid, but also perhaps, well, water, of course, for Tamás, but maybe glycogen, just carbohydrate stores. To what extent can we link it from I would say slaughter characteristic to nutritional aspects of body
Maybe
composition?
I can
Sure, go ahead.
answer first for NIRS. What is interesting for NIRS is that we don't have, because for example in DXA with Claudia, she said we have all these results and we cannot have the raw data. In NIRS, we have the raw data, so it's good point, I think. But after we have to try to find some correlation with the good parameters that we would like to analyze. So we may develop over equation to predict something and to see if they are good or not.
For CT, I think it's very difficult. So we can measure protein because it's related to the lean and water content. But what it measures CT is differences in density. So if glycogen or these components produce changes in density, it would be possible to use these CT values as a proxy for them. But I think it's really, really difficult that.
Claudia, Tamás, any
Yeah, I
thoughts on
agree.
this?
I agree with Maria.
Sorry.
Yeah.
So then we still have to revert to
Yeah.
chemical analysis and do the hard work that way.
Yeah.
Okay. I just wanted to add that for DXA, we tried to predict, for example, calcium and phosphorus or
from bone, yeah.
Yeah, but I think here it's similar as Maria said. It relies on a constant relationship between, like in bone tissues, we know that there is that the content of phosphorus is constant in bone tissue, regardless of the individual.
Yeah.
This is what we can do.
Any questions online? Please raise your hands. If not, I have a question for Örs. You said for some of this, for many of these imaging techniques, you need to have a non-moving animal. When you intubate an animal for respiration, basically it's not breathing at all. I mean, there's no muscle movements at all.
Yes.
And is that a problem? Örs, can you hear the question?
So was it about the animal is not breathing during the
Yes.
whole examination?
Yeah.
Well, the animal is breathing for the examination, but we are all the time in contact with the operator. And when they are saying, okay, please stop breathing, then we can stop the breathing, because normally for cardiac MRI, we are using rocuronium or trocrium or something neuromuscular blockade. So we are doing artificial breathing from outside of the operation room. So we have a special tunnel installed in the wall of the MRI room, where we can lead the tubing in, and for approximately 6-7 meters long cable or breathing tubes, it is possible to artificially do the pressure control breathing of the animals. And when they say, please stop the breathing for 20 seconds or 25 seconds, then we stop the breathing. And we always check the ETCO2, so the capnograph, if the animal is going back to the normal level of CO2 in the blood. And then when we say, okay, you can follow the next, you can go to the next step, because then we apply a little bit longer breathing phase. And then we say, okay, you can go for the next step, and we can stop the breathing again.
But I mean, when you say stop breathing, does that mean that the carcass is, the animal is still, the thorax is still moving? I mean, is that from?
No, because the neuromuscular blockade means that if you are don't using artificial breathing, the animal can't breathe. So this is why this method is only applicable in a deep anesthesia, because then it would mean that the animal would recognize as a suffocating if it cannot breathe but want. So this is why it is only allowed to perform in anesthesia. And if you're using this neuromuscular blockade, you have to do artificial ventilation all the time. So this is why it is not possible if you don't have a respirator, but you're using TIVA, for example, so total intravenous anesthesia. So don't try to make it without a professional respirator operator.
No, of course. Another question for several of you. Several of you talked about calibration of machines to, and Maria talked about it in different countries, because, and I was wondering, that makes it rather complicated, even for, I mean, for regular carcass grading systems, because is it the machine? Is it the animals? What is the important aspect of that calibration?
When we do the calibration of devices, we do it with 120 carcasses. In Spain, we produce 40 million per year.
Yes.
But these 120 need to be representative of the Spanish pig population. So
but that's just for Spain,
yes,
and you make equations then for
yes, and when France do the equations, they need to choose these 120 carcasses need to be selected according to the French population, either the global one or the regional one. That depends on what you want.
But in France, there's also a CT scanner that can be put on the truck. It's the machine. That's calibration is not important for the machine. It's important for the pigs that are used in that country, because Cecile mentioned that, well, the NIRS scanning is really machine dependent. Is this the calibration that you do, is that specific for the country because the carcasses are different, and then the way, or is it machine specific?
No, it's because of the carcasses are different. But this is a good topic to discuss, and we have been discussing a lot in Europe. Because now we have a lot of formulas. In Spain, we have 6 devices authorized, so we have 6 different formulas. You have seen that some devices are common in different countries, but the formulas are different. This is a discussion. It's never ending discussion, but what I can say is that's why they have, there are strong regulations to follow of the process of calibration, because it's very dependent on the technicians. The dissection, for instance, there is an error of 1.9, depending on the technicians, difference of 1.9% of limit percentage. This is quite a lot. When we use the CT, we need to calibrate it with the dissection. So the CT, the error is lower, is less than 1, but it's calibrated against dissection. So at the end, this is a good discussion, Jaap, but I cannot answer you.
I'll take two questions from the chat. What are the limitations of using NIRS on live animals versus post-mortem samples in terms of skin reflectance interference? How does the accuracy of ultrasound for determining intramuscular fat, marbling, in live cattle or pigs compare to traditional chemical analysis?
I may answer to the first part of the,
yes,
of the question.
Go ahead.
The second one, I don't know. So in this PIGWEB package, we wanted to work on carcass, and when we are thinking to the experimental trail, we are thinking that it could be nice also to have this data on live animals, because we speak more and more about phenomics and to have some specific data of animals. So I wanted to try, and what we had to do, because we have to remove the hairs of the animals, because otherwise we have some bias in the spectra. And we took some spectra actually in the ham, in the shoulder, and also in the ear, just to have a signal. But what we wanted first was to estimate, to predict the carcass composition. And for that, it was not good. the results were of bad quality, because you have to imagine that for NIRS, the signal will just penetrate in the sample of several millimeters. And so we don't have enough information from the carcass quality with this method. So we tested, but we also have a signal, but we don't know exactly what to do of this signal. So I don't know if I have answered to the question, but there are some elements of answers maybe.
Okay. And what about accuracy of the measurements, ultrasound or other, compared to chemical analysis, which is really I would say the gold standard to do it? Was the marbling or intra? Are we very accurate with our measurements?
I cannot tell you exactly the accuracy, but it's the only method that you have to evaluate intramuscular fat in live animals, the ultrasounds. So you can see, I think the accuracy is not bad, but it's not perfect, of course. In carcasses, you can use vision techniques. In fact, there is a device that is used especially in US slaughter plants. In the cat, they take a vision of the loin and they measure the intramuscular fat by vision. But in live animals, as far as I know, the only available is ultrasound. And well, you need to use it if you want to have it. But I cannot tell you exactly the accuracy.
Okay.
It's easier, I think. It must be easier, I guess, in beef than in pig, because the intramuscular fat is more visible.
Okay. Valerio has a question, interesting question. If the classification systems have changed the carcass composition, because the way we measure it and value it.
If you pay,
is it the system or is it the biology that has has the system changed the biology or the biology?
It's the biology. In fact, the devices need to be calibrated every maximum 10 years because the population change. And for sure, you know that the pig lean meat percentage of the pig has increased a lot in the last years. So this has changed, of course, the quality of the hams of the whole carcass. But to do, for instance, they ask about cured ham in Spain. To do cured ham, you need a minimum of fat. So some animals maybe are not suitable for this purpose, and you need to have a minimum of fat. It has changed, yes. If you have done genetic selection to increase the lean meat percentage, it has changed.
You need time, basically, just to let the animals get older. Yeah. Is there anybody who wants to make some other comments or question to the speakers you would like to raise? Because it's 2 minutes before 12, so we have to stop. No? Okay, then I would like to thank all the speakers for their contributions and the audience for their participation. There was one question about whether it would the recording will be available. I think, Maureen, I think it will be available at some point in time from some point in time. Yeah, it's Maureen indicated that it will be in, follow us through LinkedIn and other, but the presentations will be sent to the attendees. And I don't know about the recording itself. If it will be made accessible. Perhaps if it can, we will try to do that. So with that, I thank you for your participation. I hope you have a wonderful afternoon. And for the speakers, please stay online so we can have a short talk. Yeah.
If I can add something,
Sure.
I would like
Absolutely.
to thank you, Jaap, because you were the leader of PIGWEB, and so you give us the opportunity to work together. And I
but we did it together.
Yeah, thank you, Jaap.
Yeah.
Thank you.
And I think
Thank you,
Cornelia
Jaap.
is here also today, so thank you for because
Yes.
you were the leader of board package 6
Yes.
and so it was a work-up.
I don't know if she's back in Germany or still in South Africa, but
Thank you, Cornelia.
And it was a nice opportunity to work together on the same animals. So it was a really a European project,
absolutely.
I think.
Great. Thank you very much. Okay. Have a nice afternoon.